So as we mentioned before, bicoid, nanos, and OSCAR, all these messenger RNA are transferred to the cytoplasm of the oocyte by the nurse cells. And during the formation of this anterior-posterior axis of the oocyte, we have a cytoskeleton arrangement followed by an increase in the volume of the oocyte, as we mentioned before. And according to the different carrier protein, we have a movement, okay, of the bicoid towards the anterior side to become the most anterior determinant gene, and nanos and OSCAR bound to each other. They will be located at the posterior side of the oocyte to be the main posterior determinant. Once the anterior posterior axis of the oocyte is already formed, the nucleus will move from the posterior location to another location near to the future dorsal side of the oocyte, in order to start establishing the dorsal ventral axis of the oocyte. So as we can see, the anterior posterior axis is formed before the formation of the dorsal ventral axis. So how the dorsal ventral axis is formed? So once the nucleus, okay, moved to this location, this nucleus also at this location will secrete gherkin, okay? And this gherkin expression will form a kind of crescent shape. It's like a crescent shape. So gherkin will be expressed. It forms a kind of crescent, okay, between the nucleus. Let's say the nucleus is right here. This is gherkin, and gherkin accumulation is present between the nucleus and the membrane of the oocyte. This is the membrane of the oocyte. So as we explained before, during the anterior posterior axis formation, here the same will go for the formation of the dorsal ventral axis, since gherkin will diffuse and it will bind to its specific receptor present at the surface of those neighboring follicle cells. The same receptor is torpedo, the receptor of gherkin. And you see here, this is an egg chamber. You see the nurse cells, the follicle cells, and this is the oocyte, okay? Let's say the nucleus is right here. This is the membrane of the oocyte, and this is gherkin, okay? Gherkin will be secreted by the nucleus, and then it will diffuse to the surrounding follicle cells dorsally, okay? In order to bind to torpedo. And this gherkin diffusion, okay, when gherkin reaches those follicle cells and when it binds to the receptor, this will make a modification in the shape of those follicle cells. So usually follicle cells, you see, they are small and cuboidal in shape. They have a cuboidal shape, so that but here, after the diffusion of gherkin, this will change the morphology of those follicle cells from cuboidal to more columnar cells. So these cells, they are more elongated. They become columnar, okay, due to the impact of gherkin. But here you have to note that we don't have a long range diffusion of gherkin. You see, it's a short range diffusion of gherkin, the one in yellow here, okay, short range diffusion, and then they change the shape of those cells, okay, in order to form or after, we call them after they change their shape, we call them dorsal follicle cells, okay, as you can see. Dorsal, these are the dorsal follicle cells, so that the opposite side will be ventral, okay. In some experiments done, they tried to make maternal deficiencies. It means maternal mutations of gherkin, let's say. In the oocyte, they mutated gherkin or its receptor torpedo. And in both cases, they obtained ventralized embryos. What does it mean, ventralized embryos? It means embryos which are all over ventral. So these embryos cannot develop the dorsal side. Okay, they cannot develop the dorsal side. They are all over ventral. So this will cause the ventralization of this embryo, and this means that gherkin and its receptor torpedo are very important for the formation of the dorsal side of the oocyte, and later for the embryo after fertilization. So now I will explain the formation of the dorsal ventral polarity in Drosophila. So how this dorsal ventral axis is formed at the level of the oocyte, and it will be maintained for the whole life in Drosophila. So at the beginning, as we explained, the nucleus will move from the posterior side towards this location, okay? That will become the future dorsal side. At this location, the nucleus will secrete one protein called, or it will of course produce one messenger RNA called gherkin. This gherkin are translated into gherkin protein and bind to torpedo, the receptor present at the surface of those follicle cells, in order to dorsalize or to differentiate these follicle cells according to the dorsal morphology, so they become columnar cells. So here you should know that gherkin only diffuses to these neighboring follicle cells. So the diffusion of gherkin cannot reach the opposite side, that will become the ventral side. Okay, so gherkin is present only dorsally, and its presence will inhibit one protein called PIPE. Okay, but ventrally gherkin is absent, so PIPE is active. It can be produced, and this PIPE can activate a very long pathway, okay, in order to form the ventral side. So what I will explain now will happen just right here in this very small space between the epithelial cells and the membrane of the oocyte. This very small space is called perivitelline space. Perivitelline space. Okay, the space which is present between the membrane of the oocyte and the follicle cell. So this is the perivitelline space. In this perivitelline space, PIPE will modify one factor that we don't know exactly what is this factor. They are still studying it. They called it X. PIPE, they modify X. So we have a modified X with noodle, okay, noodle. These two factors, PIPE and noodle, will act on so noodle and this X factor will interact. Okay, we have an interaction between noodle and this X modified factor to split gastrulation deficient protein called GD, for gastrulation deficient. It's called gastrulation deficient because this gene, when it's mutated, the embryos fail to gastrulate. So they call the gene according to the mutant phenotype, so gastrulation deficient. So when gastrulation deficient is splitted, so it becomes active, so it can split another protein called SNAKE, okay, and this SNAKE, after being splitted, becomes active, and then this activated part will split Easter protein. Easter protein cleave becomes active, so it will split Spätzle, okay, which is the German noodle, okay, since this embryo, when it's mutated, is similar, okay, to this form, okay, that will look like a Spätzle or a noodle form, like this, okay. You see the noodles having this form, and after the activation of Spätzle, this activated form, splitted, will bind to Toll receptor present at the membrane of the oocyte. Okay, this Toll becomes active, so it will activate PEL and tube proteins. PEL and tube proteins, they will phosphorylate cactus and they will degrade it. Okay, cactus becomes degraded, so that dorsal protein attached to cactus will be released, and then it will enter to the nucleus in order to ventralize later these follicle cells. Okay, and in this way we have the formation of the dorsal ventral axis in Drosophila. Okay, so why this protein is called dorsal? It's called dorsal, but it's responsible for the ventralization of the oocyte, okay, and for the embryo later. Okay, since when it's mutated, okay, we don't get any ventral side, the whole embryo becomes dorsal. That's why this protein is called dorsal protein.